Today's speaker is Pau Aceves. Pau joined Certara last year, and he is a pharmacist by training. He also earned a master's degree in PK/PD modeling and simulation from the University of Manchester in the United Kingdom. His experience includes work on studies for first-in-human, proof of mechanism, and PET studies. He comes to Certara with 11 years of quantitative clinical pharmacology experience in the design, analysis, and reporting of studies in a range of therapeutic areas across all phases of drug development. Pau, welcome to today's event. I'll now turn it over to him to begin the presentation.
Thank you, Suzanne. Thank you very much for your kind introduction, and thank you to the attendees for joining me in this presentation today. So, as you can see from the title today, I'm going to be talking about a slightly unusual topic in clinical pharmacology circles, and is tracer kinetics. And this is a topic that I've been discovering myself over the last few years, and on which I have been gaining more and more interest over this period. And the reason is because I think it's an underused tool that could potentially be of use to us for drug developers. And in particularly, I believe that clinical pharmacologists are well positioned to understand the science behind tracer kinetics and learn how to apply it. So, just to give you a little bit of an introduction, I'm going to talk to you as to what we typically do as clinical pharmacologists, and how can this be different to what one might obtain from a tracer kinetic analysis. So without realizing, I think that in clinical pharmacology, we are often involved in the conduct of studies where we study PK/PD under reasonably controlled experimental conditions. And by that I mean that we oftentimes don't even realize we know factors or parameters such as the dose we administer or the concentration, the time of administration. So we have a fairly good understanding of the extent of input of our drug in this case when we are doing a drug interventional study. Or if we are conducting a drug which is infused, we also know the rate of input, so the rate of infusion. And also we know the place of administration or the site of administration, so we know now the site of absorption. And you can imagine that that's different when we're trying to learn something about a biomarker, where we don't know many of these things like dose or rate of input or rate of appearance of a biomarker. So, so then we face different, less controlled conditions when we are trying to study that type of data. Also, we oftentimes conduct our studies and we using sampling from relevant compartments like blood, urine, which are fairly accessible. And we also measure in these compartments things like drug and metabolite concentrations, and can also measure concentrations of biomarkers. And oftentimes those concentrations are rapidly equilibrating or rapidly changing, which allows us to make inferences of the effects of our drugs on certain pathways. And you can imagine that that could be different in certain biomarker situations where this rapid equilibration does not happen. Also, in phase 1 type of studies, we often conduct them in healthy volunteers, which makes the assessment of PK/PD quite easy as you compare to perhaps conducting those studies in patient populations where we do not have the luxury of retaining participants in the study for a long time. And ultimately, we also often conduct studies like ADME studies with labeled drugs to understand the ADME properties of compounds. And in all those situations, we are experts at measuring concentrations and using thereby quantitative PK/PD analysis tools to the understanding of that data. And I believe that is indeed the cornerstone of drug development these days. So tools that we apply, and I'm sure most of you are aware of, are quantitative tools like non-compartmental analysis, compartmental analysis for PK/PD, exposure response modeling, PBPK, and quantitative systems pharmacology modeling, which typically relate to the understanding of changes in concentrations of drugs or biomarkers in the body. But as I said before, what if we faced other circumstances that are less controlled? So what if we wanted to understand the rates of production of a certain biomarker or the rates of catabolism of a certain biomarker? What other quantitative tools would be at our disposal in those less controlled conditions? So in that respect, I believe that tracer kinetics could be another very useful tool in our armamentarium or toolbox. So tracers are substances such as dyes or labeled drugs, such as radioactively labeled drugs, C14, as we said before, which we use in ADME studies, or stable label isotopes, oftentimes C13 labeled compounds. These, the analysis of these typically confers a number of advantages, which come from the fact that we are able to follow or trace more than one form of the same compound, and so that unravels a number of properties that we can learn, which we cannot learn with concentration measurements alone. And I'm hoping to explain this to you in the next few slides. So indeed, tracer kinetics can help us in the understanding of turnover rates of pharmacological pathways or physiological pathways. And this is indeed a technology that is very often used in sciences such as nutrition sciences and sports sciences. So they, in those disciplines, they use these type of tracer kinetic studies all the time to learn about the sort of rates or the physiological rates of various biomarkers, or to learn about the rates of physiological functions like the rate of production of glucose or of rates of lipolysis and so on under various conditions, such as under exercise conditions, under dietary restriction conditions, and so on and so forth. So there's a wealth of literature in those sciences with regards to tracer kinetics. And I think in terms of clinical drug development, while there's some instances where people have applied this sort of technology platform, it's been underused. And I think generally that might be because of people are not generally aware of it. And I'm hoping that through the presentation today, I will raise awareness of this particular topic. And I will indeed focus on the latter type of tracers, which is stable isotope tracers, because they are safe to use as they are not radio-labeled and then can be readily applied in clinical drug development easily. So just to give you an outline of the presentation, it's going to be divided in 4 sections, and I'm going to try to give you a crash course on tracer kinetics in a very few slides. And then after that, I'm going to move to some pharmacokinetic applications, then pharmacodynamic applications. And in the last part of my presentation, I'm going to be bringing it together and talk about PK/PD modeling applications, which I think brings most power to the application of tracer kinetic data. So starting with some introductory definitions, and I would like to talk to you about what do we mean by a tracer or a tracee. And a tracer is typically a substance that is added into a system to trace the activity of something else, and typically that would be a metabolic pathway. And when we add a tracer into the system, we are not necessarily interested in the tracer, but we are interested in learning something new about what we call the tracee. The tracee would be, for example, an endogenous substance like a protein, DNA, a lipid, and so on and so forth, whose disposition, kinetics, or properties we are interested in discovering. But to do this type of studies, we need to make a number of assumptions and assume some properties for the tracer and the tracee. And I'm going to summarize them here for you. So tracers have to be indistinguishable from the tracee by the biological system. With that, I mean that when we add the tracer into the system, the biological system has to treat it in the same way as it would treat the tracee. So if it's metabolized by cytochrome P450s at a certain rate, we would expect the tracer and tracee to undergo a similar process or the same process. Also, the tracer must be detectable and distinguishable by the observer, so we must be able to label it in some fashion so as to distinguish it from the tracee, and that's where the C13 labeling, which is often used, comes into application. Then the tracer should also be introduced into the system in quantities that are small enough so as not to result in perturbations of the system that we're trying to measure. So, for example, we know that if we administer large amounts of amino acids, we can then interfere with the rates of synthesis of muscle mass, for example. And if we were interested in measuring the rates of muscle mass formation by a given pathway, we would not want to give very large amounts of amino acids so that the rates would change. So in that respect, we have to keep the quantities small enough. And typically, as a rule of thumb, people use 10% of the enrichment to be attained for that. Also, we assume that there's an instantaneous mixing of the tracer in the pool and that the pool size remains constant. Ultimately, we also assume that there's no recycling of the tracer within the pool. So now moving on to my sort of example of tracer kinetics with fishes. So here I would like to explain to you, and I believe the main principle of tracer kinetics, and I believe the key to understanding this is to understand the difference between fluxes and concentrations. And I try to illustrate this with these fish tanks. And you can imagine that these two fish tanks would be analogous to two different biomarkers that we're trying to learn something about. So in this example, let's assume also that we do not know in these systems what is the rate of appearance of the orange fishes or the blue fishes, and we don't know the rate of its appearance. And we also do not know what is the pool size, so it's the total amount of fishes in each tank. But we, however, do know based on first principles that the rate of this appearance at steady state is equal to the rate of appearance. So that's something we do know. So let's do what we typically do in clinical pharmacology, and is to take a concentration measurement of the fishes. So if we sampled from the tank at the top and at the tank at the bottom the same volume, we would reach the conclusion that we have the same concentration of fishes. That is, that we have 5 fishes per unit volume in both tanks. Now, moving on to the tracer kinetics experiment aspect of my example here, which is called the tracer dilution method. So let's imagine for a second that we could take some fishes which are identical to the fishes in the tanks and label them in a different color. And then let's assume we could incorporate those exogenously into our tanks and then take a concentration measurement. Again, we would reach the same conclusion in terms of concentrations, where we would see that we have the same concentrations of fishes at the tank at the top and at the tank at the bottom, so 5 fishes per unit volume. However, we could also readily see that there's a difference between the two tanks, and that is that the tank at the top has a ratio of 3 to 2 fishes, or a higher tracer to tracee ratio compared to that of the tank at the bottom, with only 1 labeled fish in our sample. Now, obviously, based on dilution principles, the conclusion would be that in the tank at the bottom, we have a faster flow of fishes, or a faster flux of fishes going on. Therefore, there's a higher dilution in fishes at the tank at the bottom. This would obviously give us some information indirectly through the knowledge of the tracer infusion rate, as well as the tracer to tracee ratio that we can see in our sample, as to what was the original rate of appearance that we saw in our tanks. And now for completion, let me talk to you about a different methodology that is typically done in sort of biomarker research, and that is the tracer incorporation model, which is based on is a tracer kinetic approach where we administer a precursor that gets incorporated into a certain biological pathway. And I was trying to make an analogy here as to how that would relate to the fish tank analogy. And I believe that would be similar or analogous to being able to color some of the fishes that go into the tank from the source, from the place where they originate. So let's imagine we could do that. We would achieve a similar effect. So some of the fishes that would be coming into the tank would be labeled. And therefore, we could again take some concentration measurements and measure then the tracer to tracee ratio, that is the number of labeled to unlabeled fishes, to learn something about the rate of synthesis into the tanks. And then we would see that there is a difference between the rate of synthesis between the tank at the bottom and the tank at the top. Now, from this type of experiments in real clinical studies, we obtain something that is called the fractional synthesis rate, which relates to the rate of synthesis through scaling of the pool size. The pool size is the amount or the total amount of fishes in the accessible pool size or the tank, the fish tank in this example. So hopefully with this, this has given you an insight to understand what tracer kinetics is at a very high level, of course. And now let's look at some applications. And I would like to start, as I said earlier, with some pharmacokinetic applications, which I believe probably are a little bit simpler in that sense to apply or to understand. And the first case study I would like to bring to your attention is a recent example of mine where I have been working with a client to potentially look at the application of what we call the dual tracer ADME and absolute bioavailability study as a strategy to save time and money and make a study feasible in a population that otherwise would not be feasible in. This would be a study using two tracers. One would be C14 labeled compound administered by the intended route of administration. And on top of that, we would administer an intravenous dose of an isotopic microtracer. I'm going to be giving you a little bit more of details in the next slide as to how this works. But if I had to summarize, the main advantage of this approach would be that it would allow us to conduct the study in a single period, as opposed to two periods, because if you think about this, this is in fact two different studies. And by using this dual tracer approach, we would be able to conduct this study simultaneously in a single period. Now, the advantages that this confers are manifold, including cost efficiency, as subjects do not need to stay for so long, so no need to stay for two whole periods. There's no day-to-day PK variability, as all the measurements are conducted in a single day. The shortening of the study makes this approach highly applicable to patient populations who are frail. So for example, we were looking at applying this in the oncology setting, where we expected that subjects would not be able to undergo two periods of a study like this. And thereby, it made this approach highly feasible in this particular patient population. And ultimately, it would also, and that's one of the reasons why we wanted to obtain this sort of data, was to allow us to use this ADME and absolute bioavailability data to ultimately confirm the robustness or to verify the validity of our model, which was a PBPK model that we had been working on in the past. And we wanted to ensure that it was robust enough to make some simulations for regulatory submission. But first of all, let me talk to you a little bit more as to how the study design would work and what is the concept looking like. And we don't have at the moment data for this particular example that we've been working, which is published. But I have found some published data from some authors that did conduct a similar study in healthy volunteers. And essentially, this consisted in the administration of a hot material or C14 labeled drug orally. And that would represent our ADME sub-study. And at the same time, the administration of the intravenous microtracer dose and that would happen almost simultaneously. Thereby, we would have both isotopomers happening at the same time. And what we would do then would be to sample from blood and to obtain a number of concentrations or samples over time. What we would then do is to do our bioanalytics and obtain the concentration measurements from the oral dose, as you can see on the upper graph, so the typical oral PK profile, in this case from the publication I've mentioned. And as you can see at the bottom, simultaneously we could also obtain the intravenous PK profile, which would come from the microtracer dose with the C13 labeled drug. And also, thanks to scintillation counting, we could also obtain the ADME properties of the compound by measuring the routes of excretion of the radio-labeled compound. Ultimately, as I said, the purpose of this was to bring all these data together to further verify our PBPK model. And I think most people in this WebEx will know what PBPK model is useful for, but if you're not aware, I highly encourage you to go to our previous WebEx presentations, as well as our website to find out more about PBPK. But in essence, what it allows is a type of modeling that allows to build a sort of virtual human being, thereby allowing us to predict the pharmacokinetics in that virtual system. With it, we can then do virtual clinical trial simulations, and we can use those for the purposes of streamlining our drug development programs by reducing the number of studies that are required for a certain application. But not just that, it also can allow us to optimize our label by giving recommendations in terms of certain simulated scenarios using PBPK of drug-drug interactions and other multiple and of other intrinsic or extrinsic factors of interest for which we have not conducted a specific clinical trial. And we have multiple examples, successful examples of this at Certana. Moving on to my second example, and this is another personal example of mine, of being involved in a program where we were looking at replacement therapy with an exogenously administered endogenous compound. And the title of this case study is a tracer kinetic strategy approach to obtaining a rational dose regimen selection. And this particular program I was involved with was a surfactant replacement therapy. And for those that do not know, surfactant is a substance that lines the lungs and that facilitates normal respiratory function. And under certain conditions, this surfactant the surfactant production is minimized, and therefore we have less of surfactant, which interferes with normal respiration. And this is the case of respiratory distress syndrome in neonates, which typically receive surfactant replacement therapy by intrathecal bolus. In the particular example where I had the pleasure of being involved, this was a project where we were looking at a new surfactant delivery method intended for adult dosing, and where we were looking at administering this surfactant in a way which would be more amenable than intrathecal bolus, so a better delivery method in that case. And personally, when looking at this project, I could immediately see a number of complexities, and especially from a clinical pharmacology perspective, we did not have a way of determining the concentrations of the surfactant in plasma. Therefore, we couldn't obtain plasma pharmacokinetics measurements in this for this program. Also, we could really not do or obtain concentrations, let's say, from bronchoalveolar lavage samples, because we could not really distinguish between the concentrations of the endogenously produced surfactant and that which we administered exogenously. So both of them would be mixed in the lungs, and we could not distinguish one from the other. So in a way, it was really challenging to do conventional pharmacokinetics. And now and that raised a few questions in terms of how to come up with a rational dose regimen selection. And a few key questions were, well, what was the replacement dose needed to replace the loss of surfactant in the lungs for those various pulmonary diseases? What was the frequency of administration required? And whether something like disease severity would have an impact on the dose that was required for administration in the various indications we were looking at. And these questions were extremely crucial because one property was that for this particular program, duration of administration or inhalation, in our case, was related to the total amount required. So the larger the dose that was required, we had to make the subject sit and stay to receive therapy for longer. So that was really inconvenient, and from that perspective, we really wanted to optimize the dose administered. So we tried to find ways of alternative ways of coming up with a regimen dose selection. And during that process, I came to discover tracer kinetics. And I discovered it through this publication you can see here from Kogo et al. In 2007, where they studied the lung surfactant kinetics in adult patients with respiratory distress syndrome. What they did was to measure the tracer to tracee ratio, that is the labeled to unlabeled surfactant ratio, over time by taking multiple bronchoalveolar lavage samples. And they obtained as an output from that study the type of graph you can see at the bottom. So you can see that the blue line represents the adult patients with respiratory distress syndrome, while the sort of pale round dots represent the healthy controls. Now you can immediately see that there's a few things that strike us very quickly. And one is that we can see there's a slight differences in half-life potentially, or that we can determine the half-life in the two populations. So that would be one thing that would be extremely useful to the program I was talking about. And also we can see immediately another property that is conferred by virtue of studying the tracer to tracee ratio, and that is that the blue line had much higher tracer to tracee ratio than the pale line at the bottom. And therefore, the conclusion from this is that the pool size or the amounts of surfactant in the lungs of patients with ARDS was much lower, resulting in a different level of dilution between the ARDS patients and the controls. So the ARDS patients having a less diluted tracer concentration. But in addition to that, and that's something I'm going to be highlighting through my presentation, the authors from this publication further try to learn from the data they obtain by applying some modeling. And I think this is a key feature that is important to bear in mind, that to extract the maximum amount of knowledge from this type of studies, I think it's really maximized through the use of modeling. In this case, they modeled this data with a two-compartment type of PD model, where they built in the sort of rates of clearance from each of the two compartments and rates of transfers between the two. And then with that, they were able to obtain a critical piece of information. On the one hand, they obtained the rates, as I said, rates of transfer and the rates of clearance from each compartment. But I think quite importantly, they also were able to obtain an estimate of the pool size or the amounts of surfactant in the lungs of patients and controls, thereby realizing, as I said before, that the amount of surfactant in patients was much lower. Now, taking inspiration from that particular study, we devised a strategy to come up with a rational dose regimen selection. That involved the conduct of a study where we would estimate the surfactant pool size in subjects with normal lungs, as we have seen in the example before, and also in patients with the indication we were interested in looking at. By looking at the difference between healthies and patients, we would then be able to estimate how much surfactant had been lost in the patient population, and thereby learn something about the dose that needed to be replaced. Also, with the tracer kinetic data we would obtain, we would be able then to have an estimate of the half-life or even model the data to then simulate a regimen that we would then apply in a future clinical study. And then, to further complete this picture, we were planning to do a gamma scintigraphy sub-study that would allow us to understand the efficiency of delivery of our device with respect to the surfactant deposition in the lung. So with the gamma scintigraphy, we would learn how much of that surfactant would be being deposited by our device in the lung, analogous to learning about the bioavailability. Now, with these three pieces of information, we would be then able to make those recommendations for various indications in the upcoming patient studies in phase 2. Now, moving to the next section, which is the pharmacodynamic applications of stable isotope-based biomarkers. And indeed, tracer kinetics, as we have already seen or mentioned, they obviously have applications when trying to learn something about bodily substances like amino acids, lipids, even DNA, and so on and so forth. And that is because they allow us to understand the turnover rates of pharmacological pathways of interest. But not just that, potentially what I'm calling flux-based biomarkers, which is sort of the tracer to trace ratio type of biomarker data, can also be more of robust biomarkers as well. And that is specifically the case, I believe, where we're trying to learn something about the intrinsic tissue pathway activity, for example, for biomarkers that have a slow half-life. That is that it takes a long time to clear from, let's say, systemic circulation from where we are off, from wherever we are sampling. Also, in certain circumstances, we can find that concentration-based biomarkers cannot be informative in conditions, for example, like liver disease, where concentrations change not because there are changes in intrinsic sort of activity of whichever pathway we're trying to measure, but the changes in concentration reflect something else, like, for example, changes in hepatic blood flow, changes in volume of distribution, like edema, or changes in hepatic clearance. And that's something that flux-based biomarkers should be less affected by. So I think ultimately these tracer kinetic approaches could become a powerful technique specifically to study treatments that target slow turnover processes. And that was my conclusion for one of the programs I was involved with. Well, we were looking to learn about changes in fibrotic biomarkers, and you will see what I'm talking about in the next slide. But essentially, the main advantage would be that for some in the context of phase 1 trials where we do not have a long time to see changes in biomarkers, and where we're trying to learn something from a biomarker that takes a long time to change in concentrations, we could have an advantage if we were able to measure the amount of new protein or new whatever and biomarker we're trying to measure, rather than measuring total concentrations. So moving on to the example I'm talking about, we sort of basically saw these as using tracer kinetics in this context as a strategy to obtain a fast proof of concept study by measuring synthesis rates. And I'm going to tell you a little bit how this would have helped in terms of giving us a faster proof of concept. So here we were looking, as I said, at the effects on fibrogenesis from an antifibrotic drug for chronic kidney disease. And the particular biomarkers of interest were extracellular matrix proteins, which we knew took a long time to change, so they had a very slow turnover rate. So changes in biomarkers were expected to take a very long time, even up to 6 months to see any changes. Obviously, in the context of a phase 1 study, we did not have the luxury of such long times. So we devised this tracer kinetic sub-study within the first-in-human study, which involves the administration of deuterated water, so heavy water, to the study subjects, with CKD. And what we expected then was that this deuterated water would be incorporated into the non-essential amino acids of those patients, and those amino acids would then be incorporated into the extracellular matrix proteins. Then by taking urine samples and making determinations of the tracer to tracee, again by doing the tracer kinetic measurements, we would be able to determine the percent new ECM protein, extracellular matrix protein. And by doing that, as I said, we were hoping that we would have greater sensitivity to see a change in those biomarkers much faster than that which we have seen if we had measured total concentrations, which, as we said, those biomarkers had a slow turnover. And at the same time, when we measure concentrations, we are measuring the total amounts, that is new material as well as the old material that is still circulating around. And the way we wanted to implement it is specifically in the context of this first-in-human umbrella protocol, which also included a CKD or patient sub-study to obtain the proof of mechanism. So, as I said, after completion of the initial single ascending dose and multiple ascending dose parts, or almost in parallel to the multiple ascending dose cohorts, we would start dosing or treating those patients with deuterated water. And that would then start to label the extracellular matrix protein with the objective to increase the sensitivity to detect changes in those biomarkers at earlier times. Also, the ultimate goal of this exercise was to use that data generated from the tracer kinetic study or sub-study in the first-in-human to start building highly mechanistic models that would allow us then to inform dose selection for our proof of concept study. And we were actually considering the application of quantitative systems pharmacology modeling, which is a bottom-up type of modeling similar to PBPK, but apply to biomarkers or pharmacodynamics in the sense that we build these highly mechanistic models that allow us to make predictions by building enough detail into those models to allow us to make simulations with those models. And with those simulations, then we can better inform sort of the dose selection and multiple other aspects, which I'm going to be talking about in my next few slides. But essentially, our hope was that by doing this study, within 4 to 6 months, we would be able to come up with a dosing strategy, a positive signal in terms of proof of mechanism, and be able to move to a well-informed proof of concept study, thereby reaching sort of optimizing our early drug development. My last example in terms of pharmacodynamic applications of tracer kinetics is about using tracer kinetic approach to understand the brain amyloid beta synthesis rates. And this example is a published example by Kleijn et al. From 2013. And basically, what they did here was to administer C13 leucine to healthy volunteers as an infusion, and then look at the effects of a drug intervention into the rates of production of A beta. And so they looked at it in 3 different arms. So they used placebo, they used low dose, and high dose. As you can see from the concentration versus time profiles at the bottom on the left-hand side, let me try and see if I can point it to you. Okay. So as you can see here on the left-hand side, they saw robust concentration changes in CSF of this A beta protein, which relates to Alzheimer's disease. They also saw changes in the tracer to tracee ratio versus time curves. And it's something we haven't gone over yet, but typically our upslope of that tracer to tracee ratio curve should relate to the production or the synthesis rate of that particular biomarker. in this case the A beta. And the downswing of that curve relates to the clearance of that particular biomarker. And that we could see here that there was an effect as well on those particular curves of that biomarker. And again, we could see that there was a dose relationship, so with a high dose having the biggest effect in both the concentrations and the rates of synthesis. So continuing on this example, I'm going to swiftly now move to the PK/PD modeling applications, because one of our consultants here, Hub, worked on the building of a highly mechanistic population PK/PD model of the data we just seen from the previous pharmacodynamic study of A beta concentrations in CSF. And I think this is an excellent example of how to build a mechanistic model that resembles the known physiology of A beta. So on the left-hand side, I've put a diagram of the known physiology, and on the right-hand side, we have a diagram of the model that closely resembles that of the known physiology with the different steps. And the model that Hub built essentially consisted of a model with multiple steps, starting as leucine as the precursor to various proteins that get incorporated to form A beta, and thereby reflecting the biology that we've just seen on the diagram on the left-hand side. And then the model was able to accept as an input standard leucine coming from sort of regular diet, but also the leucine that was administered in the study as C13 labeled leucine. By jointly modeling those two isotopomers, as well as the concentration data, they were able to verify that their model truly supported the known A beta biology. As you can see at the bottom here, the model was able to closely explain or describe the concentration versus time changes of total A beta in the brain, as well as the fraction of labeled A beta in CSF as well. So by virtue of the ability of the model to explain both factors, so the factors that relate to the synthesis rate as well as the changes in the equilibrium, that is the static concentration measurement, then they were able to gain a closer insight as to the workings of this physiological or pharmacological pathway. In fact, their model predicted a number of things, and one of the things that was useful to predict is that the upswing that it had been seen in the fraction labeled A beta depended on more factors beyond the rate of synthesis of A beta. However, they could obtain in an indirect fashion using the model the rate of synthesis. As you can see on the right-hand side, the rate of synthesis of A beta was indirectly determined from the rate of the model that related to the production of A beta in the brain. They also were able to predict with their model that the time of administration that had been used in this particular clinical trial for the C13 leucine was not optimal, and that a different time of administration, an earlier time of administration, would have been more optimal to detect changes in the fractional synthesis rate. Ultimately, I think this example highlights the importance of combining tracer kinetic data with modeling to tease out and correctly interpret the tracer kinetic data. And I think this model would once built, is there to generate multiple simulations. So one could simulate multiple what-if scenarios that could be useful for the design of future clinical studies. And then my last case study is another modeling example. And in this case is a case of quantitative systems pharmacology model that, as I mentioned before, is a highly mechanistic type of modeling, which tries to resemble the network of a physiological network as closely as possible, so as to be useful to explain complex emerging properties of that system. And in this particular example I'm showing you here, this was a model by Lu et al., and they published in 2014. And it's a model that I find really fascinating as it was extremely useful at multiple levels. So they built the model you can see on the right-hand side. As I said, quantitative systems pharmacology model, highly mechanistic, encompassing the necessary steps to explain the disposition of cholesterol in the body and multiple other analytes that relate to the cholesterol metabolism. They did this particular model with the intention to explore two different therapeutic interventions. On the one hand, CETP inhibition, and on the other one, ABCA1 upregulation. And as some of you might know, CETP inhibition was a therapeutic approach that was tried a number of years ago, where multiple clinical studies from phase 1 to phase 3 were conducting with a very high expenditure to ultimately reveal that this type of therapeutic intervention did not result in positive or noticeable cardiovascular outcomes. So this model was a bit of a retrospective exercise to try and understand why CETP inhibition might not have worked, and also trying to understand whether ABCA1 would work. Another thing I would like to highlight from this example is how many of the parameters that they used for building the model originated from tracer kinetic studies. And I've highlighted that on the right-hand side by circling the steps in the model that used some kind of tracer kinetic data, either directly or through some transformation, therefore highlighting the importance of actually having that tracer kinetic data to build this type of models. I also really like from this example the fact that they verified their model by conducting a virtual tracer kinetic study. So without using any tracer kinetic data in that sense, they simulated a tracer kinetic study of IV-administered ApoA1, and they overlaid the predictions from their model, so their simulated data, on top of the observations from a clinical study, which had not been used for building the model. The outcome of this verification was that their model was very much able to predict the observations from that clinical study, thereby giving greater confidence in the ability of the model to make predictions. Ultimately, I think the extremely high value of this model was that it was able to predict that that CETP inhibition had a lack of an effect on what we call the reverse cholesterol transfer rate, which is the rate of clearance of cholesterol from peripheral tissues out of the body, or the elimination of cholesterol. And while it did predict that CETP inhibition could cause perturbations and transient increases in HDL concentrations, ultimately it did not exert an effect on this reverse cholesterol transfer rate, and that would explain why no effects were seen on cardiovascular outcomes. In addition to that, the model was used to evaluate some candidate biomarkers for ABCA1 activity, which was also useful to inform their next steps for drug development. I think this case really case study or this publication really showcases the importance of combining tracer kinetic data together with quantitative systems pharmacology in drug development, and especially if you do that early on. So just to end my presentation, I would like to give you some final remarks. And I would like to highlight that using tracer kinetics in clinical studies is relatively a straightforward regulatory pathway, because there's extensive safety data of the use of stable label isotopes. Stable label isotopes have been used for more than 8 decades and for the conduct of clinical trials. Also, as you might have sort of gathered from my examples, is that stable isotopes can offer, under certain circumstances, the drug developers a unique quantitative tools And I'm just going to now go over some of the benefits that I've mentioned before, but I think it's important to highlight them again. And as we've seen through the various examples, through the use of tracer kinetics, we were able to optimize and make feasible the conduct of ADME and absolute BA study in a population like oncology patients, where we could then use that data to further inform our PK model to optimize our label and also to streamline the number of studies required for our drug development. Also, we've seen an example where tracer kinetics would allow us to differentiate between exogenous and endogenous substances, and thereby allowing us to come up with a dose selection strategy for a replacement therapy compound. It also allows us to potentially accelerate or to get a faster POC by obtaining an early proof of mechanism study in the context of a phase 1 study for a biomarker that had slow turnover rate. And ultimately, and as we have seen in the modeling example that I last presented, tracer kinetics can also help confirm the mechanism of action, the likelihood of efficacy, or lack thereof, as we have seen with the CTP inhibitor example, and also use modeling approaches combined with tracer kinetic data to simulate virtual clinical trials where we could explore what-if scenarios that would be hard to undertake in real clinical studies. Also, they could allow us to, through this combination of tracer kinetics with QSP modeling, for example, to evaluate biomarkers. And ultimately, and I think that's a key, that's key to drug development, they could help us elucidate or help us select doses for our first human studies if we started working on those pieces of data very early, or if we conducted some of these tracer kinetic studies early, it would be helpful to build models to inform the doses for phase 2 dose selection. So with that, I would like to end up with an analogy, and I sometimes feel that in clinical pharmacology, we're a bit like the ship surgeons of clinical drug development, in the sense that we have multiple tools that we have at our disposal, and that our job is to select the right tool for the right job, or the right tool to treat the right disease. And while tracer kinetics might not be a tool that you use in your day-to-day work, so it's not your hammer or your hacksaw, tracer kinetics could potentially be that rare tool that, if you're aware of it, it could be extremely useful to help you in your drug development efforts. With this, I would like to acknowledge a few people that have contributed to the presentation and thank them. And also, here I have some references as to the studies and case examples that we've been going over today. And with that, I would like to open the floor for questions.
Okay. Thank you so much, Pao, for that very thought-provoking presentation. We'd now like to encourage our audience to submit their questions to Pao in the Q&A box. Let's see what our audience wants to know about. It looks like our first question, Pao, is someone wants to know, is the fractional synthesis rate a direct measure of synthesis, and can you give us a definition?
Not really. No, it's not a direct measurement of synthesis. Let me go to a different slide. One moment. Okay. So basically, the definition of fractional synthesis rate is that is the percent of trace pool synthesized from the precursor. And as you might remember, the formula to obtain the synthesis rate from the fractional synthesis rate is to multiply the FSR times the pool size being the total amount of that particular substance of interest. And the fractional synthesis rate, as I mentioned, is obtained from the upswing of the tracer to tracee ratio curve. And you can see here on the bottom of the slide a graph of tracer to trace C labeled A beta again, and from that initial upswing we can determine the rate of, or the fractional synthesis rate. Now, if we know the pool size, or we have an indirect way of estimating the pool size, then we can obtain the synthesis rate indeed.
Someone wants to know, do you see a case for applying the dual tracer approach in healthy volunteer studies?
Yeah, that's an interesting question. In fact, the example I've shown in my slides comes from a healthy volunteer study. My personal view on that is the case for using the dual tracer approach. Let me go back to that slide just to remind people of that particular case study. I think based on costs of conducting the microtracer sub-study might outweigh the sort of benefits of doing this particular type of approach in healthy volunteers. Now, I think that should be evaluated on a case-by-case basis. So there may be a case where you know, when one might be able to use LC-MS-MS to determine the IV microtracer PK curve, and then it may be less costly than using AMS, and thereby making it economically feasible. But ultimately, I see more of a case for using this approach in studies where one can only use patients.
Looks like we have one final question from our audience. Someone wants to know, was there any evidence that the pool sizes determined by tracer kinetics had any resemblance to reality?
Yeah, that's a fair, a valid comment, and that's something I was concerned about in my example of the lung surfactant kinetics. So as you might remember, they derive the total amount of surfactants by estimating it indirectly through modeling, through applying a two-compartment model to the data that they observed in this particular publication. Now the concern would be whether that pool size has any resemblance with you know, what a real measurement might be. I think if we go to one of my backups, yes, so basically here we could see they saw indeed that obviously there was significant differences in the pool size in ARDS patients in yellow versus control, which makes sense based on what is known about the disease. So it was expected that they would have a lower amount of surfactant in their lungs, this particular patient population. Now, is that absolute amount really reflecting that which we might find in reality? And they found there was some indirect evidence that that was indeed the case, and they looked at publications where surfactant had been measured in cadavers, and in a publication with from Revello et al., they could compare the differences between the actual measurements, direct measurements of surfactant in the lungs of cadavers versus that obtained through tracer kinetics, and they observed that they were highly compatible, thereby giving further evidence that the estimates of the pool size was actually relevant in that sense. And that was one of the main reasons why I thought this was a highly interesting example to our particular case study, where we were trying to learn about the amount of surfactant that was required for replacement in those patients with various pulmonary conditions.